Hello everybody. Uh, Today we are going to look at the first animal group, porifera. The name porifera indicates that these animals are pore-bearing animals. They are the most primitive group of animals. Although we see these animals as multicellular, however, they are still on a cell grade organizations. We don't find any tissue in this animal. So if you look at this chart, the sponge lies the most at the bottom, and this is the most primitive because there are many features that makes this animal to be a primitive. Like when we discuss about its characteristics, probably we will, by name itself, you look at that porifera, meaning pori means lot of pores, so pore bearing animals. Cellular grade of organization, these animals do not have any tissue. However, we are going to see that world is made up of many cells. However, there is no tissue. No cells are found working together in coordinations forming tissue. Therefore, these animals is a cellular grade organizations, and it is multicellular. The animals is asymmetric because the animal cannot be divided into any equal halves. These animals are sedentary, it doesn't move. These animals in fact was being classified as a plant until a few years back. However, today it is known that it is the most primitive animals. These group of animals have made up of many cells. However, they don't have a true tissues. Therefore, these animals are called porozua. Please understand that these group of animals do not have a true tissue. Therefore, although they have many cells, and they are known as porozua. This group of animals, although it's a quite a small number, around 4,000 different species of these animals are known, and they play a significant role in the ecology, and therefore these phyla is considered as a major phyla. Let's look at some more specific characteristics, like these animal has got diploblastic, meaning diplo means it has got three germ layers: ectoderm, endoderms, and mesoderms. But this animal has got two layers, body layers, and ectoderm and endoderms. Endoderms are made up of choanocytes, which we are going to discuss about it. These choanocyte cells are very important for this group of animals. There are a lot of pores called ostia. They have got spongiocele, which is big vase like animals. It has big openings called ostia, and these animals' bodies are being supported by a calcareous spicules. Calcareous spicules, or the made up of the spicules that are made up of silicones. This group of animals has got a high power of regenerations, and it also forms some budding off. It buds off and forms some animals. So the reproduction can be sexual and asexual both. Development of animal is either direct or indirect. Direct meaning it can reproduce similar kind directly. Indirect is when these animals produce a larval form, and this larval form slowly change into adult. Such development are called indirect development. If you look at this, is how the animal features look like. Osticulum is an outer opening, and there your body of this animal has got a lot of pores, and this is called ostium or the pores. And within this diploblastic layer, diploblastic body wall has got a lot of canal system where the water can pass through into the body and it comes out of the osticulum. Alright, as I said, these animals has got many cells, and cells are of different types. One of the major type is choanocytes, which helps the animal to trap the food and also to take in the water from the pores. So these choanocytes lines up in the inner endodermal cells. Endodermal cells are all made up of choanocytes. These choanocytes has the collar cells and also the flagellum. The movement of the flagellums helps to pump out the water out of the osticulum. As a result, the water enters through the ostia from the body pores. There are three kinds of animals has been classified into three classes. There are four types of four different classes: calcareal, hexatenilidia, spongia, and sclerospongia. So these are basically the type of materials what the spicules are made up of. This classification is based on the substance with which the spicules are made up of. Calciums, if it is made up of calcium, then that group of this porifera are called calcareal. And silicon, sorry, not SCI, siliceous silicon materials makes the spicule, that is hexatenilidia. Demospingia, either of or non scleridia, both are embedded in the same animals. These are the true details of calcarea. You are going to see how this is made up of. These are the types. You can look at some more characteristics on that. These are two different orders under the calcarea. Hexatenilidia is the where you will see these siliceous spicules, and we have two different types of sponges that we see. Euplectilla is the one that we see in our laboratory. Demospingia, again, as I said, is either are absent of the skeletons. So there are three types of chondrite. There are four different types of orders, and then the one that we are going to study is the Spongilia or Chelina, or we are also see the Euspongia. So these are the two different orders. Probably we will study in our practicals. Sclerospongia, both the type of are found in this, and these are the examples are acanthocytes or astrosclera or stromatospongia. So these are quite a most primitive type of these does not have any. These has both the spicules, however, is found both calcareous as well as the siliceous spicules. Sponges are also divided based on the type of canal system in the body, like ascon type, cycon type, and the leucon type. So basically here, looking at the complexity of the animals, there are two types of cycons, ascon and cycon. Ascon is the most simplest and primitive, and the cycons are more complex. So these are the different types of cells found in these animals arranged over one another. The ectoderms are all made up of arcucite, scleroblasts, collencyte, colonocytes, amebocytes, and porocytes. The inner wall of this body of the sponge are made up of poenocytes, which help to suck in the water, and it creates a current, and then from the osculum the water is being expelled out. So this is how the inner wall is made up of poenocytes. This blue one, poenocytes. The outer one, these are the spicules, and these are the sclerocytes, and these are the pinocytes. And there are pores here. On the pinocytes there are pores, and these are amoebocytes, right? And these are the different types of cells that are found, and that's why they are heaped on one another and never has formed the tissue. This animal has got different types of skeletons, monoaxon, triaxons, and tetraxons. If the pointing end of the spicule is only one directional, then it's monoaxon. If the pointed spicules or the spicules grow in 3 directions, they are called triaxons. If this grows in two directions, diagonal, it's a 4, is a tetraxon. If it's a more than 4, it's a polyaxon. Now these are basically these animals has got a canal system where the water can pass in from the pore into the spongocoel and the osculums. The water moves from here, from outside to into the body, and then comes out. And basically these water currents carries out the following function. One is respiration, for breathing purpose. Nutrition, it brings in the food along with the water current. Excretions, it throw out the waste what is produced in the body. And reproductions, these gametes are being freely passed into water, and then it moves and gets fertilized. So based on this canal system, the sponges are divided into 3 types: ascon type, sycon type, and leucon types. With that, thank you.